Hello, this is a COVID-19 video that seeks to look at where we are now as of April 2021. This is an informational video, so please seek medical advice from a medical doctor. 15 to 16 months after the first report of the SARS-CoV-2 virus, 151 million subjects have tested positive for SARS-CoV-2 virus globally. Of this, more than 3 million people have died of COVID-19 or its complications. These deaths do not include those who died of other causes due to the inability to access healthcare services during the COVID crisis. This pandemic has posed a great menace to human physical and mental health on a global scale. This video will review what is currently known about COVID-19, available treatments and vaccines, as well as possible therapeutics widely used by the masses but not recommended by authorities. This slide describes COVID progression once an individual is exposed to the virus. Following exposure to SARS-CoV-2, individuals go up to 5 days without showing any symptoms. During this phase, the virus is multiplying rapidly and may be detected using PCR nasal swab tests. Some individuals begin transmitting the virus to unsuspecting people that they are interacting with, such as family members. Once the virus reaches a maximum, individuals exhibit mild to moderate illness. During this time, the immune system is producing antibodies to help the body fight the infection. Symptomatic individuals experience increasingly severe symptoms such as diarrhea, poor appetite, nausea, vomiting, confusion, headache, chest distress, fever, cough, shortness of breath, sputum production, myalgia or fatigue. If the symptoms do not resolve, individuals are hospitalized due to shortness of breath, requiring oxygen supplementation. Some individuals progress to acute respiratory distress, culminating in ICU admission due to inability to sustain respiration. Acute respiratory failure is the hallmark of severe COVID-19. Reversal back to health can and does occur at each stage of the disease. However, comorbidities such as hypertension, asthma, diabetes, obesity, or genetic mutations may accelerate progression of the disease, ultimately leading to death. The SARS-CoV-2 virus spreads through airborne transmission or by saliva droplets, and sometimes possibly by fecal contamination. The virus is detected in sputum or saliva, mucus, blood, and fecal matter. The most common route of exposure is the respiratory system. Upon exposure, the SARS-CoV-2 virus enters the body via the nose or mouth, travels down the throat into the bronchus and bronchioles, ending up in the air sac or alveoli. SARS-CoV-2 uses the spike protein to gain entry into epithelial cells via a protein known as angiotensin-converting enzyme 2 or ACE2 receptor, and the transmembrane protease serine 2 enzyme. The virus enters the cell within 10 minutes and releases its RNA, which is the genetic material. The viral genetic material takes control of the human cell and begins replicating and translating the RNA into new RNA and proteins respectively. The newly synthesized proteins and copied RNA are processed, packaged, and released into the alveoli within 10 hours. Once there are 1,000 virions or virus copies in a cell, the host cell lyses itself and releases the virions to surrounding tissues. Once the virions are released from the alveolar type 2 cells, they are detected and destroyed by immune cells, that is the macrophages and the dendritic cells, which are the major first line of defense in the lung. The immune cells produce proteins and chemicals known as chemokines and cytokines to recruit other immune cells to help fight the virus. The cytokines, e.g., IL-6, diffuse out of the alveoli into the pulmonary capillaries. In the capillaries, they enable the migration of other immune cells such as neutrophils and monocytes, as well as other protective cells like fibroblasts that repair and maintain the alveoli from the capillaries and the interstitial space respectively. Both neutrophils and fibroblasts can contribute to the alveoli damage by releasing reactive oxygen species and reactive nitrogen species, and as well as inducing scarring in the lung. At the same time, the virus uses the same pathway to enter the blood capillaries, hence allowing SARS-CoV-2 to move from the lungs into the rest of the body. Once the virus enters the capillaries, it travels to the rest of the lung and body. When the virus encounters other lung cells, it will infect the cells and produce cytokines. The virus also triggers reactive oxygen species, which activate cytokine release through other mechanisms. Both pathways lead to a cytokine storm, which leads to acute respiratory distress in the lung. ARD may also be followed by a bacterial infection that can lead to sepsis. In sepsis, immune response to infection overwhelms the body by releasing chemicals into the blood to fight the infection. Those chemicals trigger widespread inflammation, which leads to clots, blood clots, and leaky blood vessels. As a result, blood flow to the organs is impaired, and that means the organs do not receive oxygen or nutrients, leading to organ damage and/or death. In the worst cases, blood pressure drops, the heart weakens, and the patient goes into septic shock. Once this happens, multiple organs will fail, including the lungs, intestines, brain, heart, liver, kidneys, eyes, and patients who recover may return to normal. However, some may become long haulers. Long haulers continue to experience COVID-19 symptoms for up to 1 year post-COVID recovery. Some of these long haulers from COVID-19 have been known to die while still waiting to recover from COVID-19. In the US and elsewhere globally, individuals with cardiovascular disease, hypertension, metabolic disease, and obesity were more likely to be hospitalized with COVID-19 than those with other comorbidities. Among children, only children with obesity were likely to be hospitalized and die from acute respiratory distress syndrome. Not surprisingly, increased ACE2 receptor expression is observed in lungs of individuals with type 2 diabetes, in individuals who have chronic obstructive pulmonary disorder, in smokers, and in the hearts of people with cardiovascular disease, providing a basis for severe outcomes in people with cardiovascular disease and type 2 diabetes. In addition, in these individuals, there's a high neutrophil count and a low B and T cell count, which is independently observed in people, in older individuals, in people with diabetes, people with hypertension, and people with morbid obesity. In children, there were more children with no known condition that suffered severe consequences or were hospitalized with COVID-19, and there was nothing attributed to the high numbers observed in children. Up to April of 2021, more than 5,000 clinical trials had been registered. 1,035 of these were registered in the US and have been completed globally. Throughout this period, thousands of virus and host-based drugs have been tested. A whole arsenal of drugs, including newly developed and repurposed drugs, monoclonal antibody therapies, convalescent plasma, and vaccines have been tested in these trials. However, very few of these have received recommendation for use or have gained emergency use authorization. For outpatients who have mild to moderate symptoms of COVID-19, there are no recommended treatments. Only supportive measures and symptom management are indicated for these patients. These patients will include adults with COVID-19 that visit a clinic or a doctor's office, adults with COVID-19 discharged from the emergency department, and adults with COVID-19 following inpatient discharge from hospitals. These patients should not take chloroquine, hydroxychloroquine with or without azithromycin, or lopinavir, ritonavir, and other HIV protease inhibitors. For outpatients with a risk of disease progression, only monoclonal antibody therapy is indicated. 4 of these are neutralizing monoclonal antibodies that recognize the receptor binding domain of SARS-CoV-2 virus and deactivate it. The antibodies are used in combinations of 2, bamlanivimab plus estesavimab, and casirivimab and imdevimab. However, these antibodies are at risk because the SARS-CoV-2 is mutating rapidly. In fact, the FDA has revoked the emergency use authorization for bamlanivimab and is closely watching other monoclonal antibodies due to increasing resistance to observed in the SARS-CoV-2 variants. In hospitalized patients, the new antiviral drug, Remdesivir, is moderately successful in hospitalized patients that do not require oxygen supplementation but are at a high risk for disease progression. It is also given as an add-on therapy for patients requiring oxygen supplementation. For hospitalized patients who require supplemental oxygen or those requiring mechanical ventilation, only Dexamethasone, a repurposed host-based drug, which is a steroid, is approved for use. Tocilizumab, a monoclonal antibody with activity against pro-inflammatory cytokine IL-6, has also been approved as an add-on therapy, only for hospitalized patients requiring oxygen supplementation or ventilation. It is not efficacious by itself. More earnest investigations are ongoing for development of preventative agents. Notably, Sanotize, a nitric oxide nasal spray, has just entered phase 2 clinical trials. Invisimask, a nasal spray containing a neutralizing monoclonal antibody, and Covivac, a single-dose intranasal vaccine against COVID-19, are just about to start clinical trials. In addition, although the jury is still out, Ivermectin is under evaluation for the treatment of COVID-19 and so far has favorable results in 21 completed clinical trials globally. Another treatment that is indicated is the high-titer COVID-19 convalescent plasma, which received emergency use authorization for treatment of hospitalized patients with COVID-19 early in the disease course or those who have impaired immunity. 2 other compounds are vitamin D and cannabidiol, which have also been studied extensively. Clearly, a lot of money and brain power has been allocated to COVID-19 for the last 15 months. Nevertheless, we are still in a very precarious position and literally almost at the mercy of the rapidly mutating SARS-CoV-2 virus, as evidenced by the overwhelming situation playing out in India and Brazil. At the time of preparing these slides, WHO has approved 2 mRNA vaccines, Pfizer-BioNTech Comirnaty and Moderna vaccines, 2 DNA vaccines, Covishield by AstraZeneca and Janssen adenovirus 26-CoV-2 by J&J. Last but not least, a protein-based vaccine has been developed by Sinopharm from China. FDA has approved only 3 of these vaccines for COVID-19, while the European Medicines Agency has approved 4. Globally, different vaccines are being rolled out with very slow uptake due to shortages and the COVID situation in India, which is a major manufacturer of vaccines. All vaccines are directed against the spike protein of the SARS-CoV-2 virus and are injected into the deltoid muscle of the arm. Once they enter the cell, mRNA vaccines are immediately translated into the spike protein in the cytoplasm, while DNA vaccines must enter the nucleus for transcription then move into the cytoplasm for translation into the spike protein. Protein-based vaccines are ready for immune cell activation. In all cases, once the spike protein is detected by defense immune cells, that is the resident macrophages and dendritic cells, they are engulfed and destroyed, then presented to B cells and T cells. Activated CD4 T cells interact with activated B cells, facilitating the conversion of B cells to mature plasma cells that produce antibodies. Activated CD8 T cells are cytotoxic and destroy infected cells. Additionally, memory T and B cells are produced for future defenses. A second dose for Pfizer, Moderna, AZ, and Sinopharm is required to amplify the B and T cell signals. Currently, there are over 100 vaccines in clinical trials and 184 vaccines in preclinical trials. So the war is still on for vaccine development, and pretty soon we should have more to choose from. So in this slide, I wanted to discuss the vaccine manenos or vaccine troubles that include cerebral sinus thrombosis or clots that have been described in the clinic for 2 vaccines particularly, but we will see later on that it's all of the vaccines that have shown signs of cerebral sinus thrombosis or clots. So more than 1.19 billion vaccine doses have been administered worldwide. That's equal to 16% of the global population. This percentage differs from country to country, with Seychelles boasting 61% of its country fully vaccinated. Vaccination against SARS-CoV-2 has resulted in a rare immune thrombotic thrombocytopenia, which is a clot characterized by decreased platelets, possibly mediated by autoimmune antibodies against platelet factor 4. The condition has been named vaccine-induced immune thrombotic thrombocytopenia. So far, moderate to severe thrombotic thrombocytopenia complications occur at unusual parts of the body, such as the brain and abdomen, and are detected approximately 1 to 2 weeks after vaccination. When first detected, AstraZeneca and J&J vaccines were placed on hold. After investigations, the EMA and the FDA found a link existed between the vaccine and the clots, but decided the benefits of the vaccine outweighed the risks. So both agencies concluded that the risk of mortality from COVID-19 is much greater than the risk of mortality from vaccine-related side effects. Furthermore, similar thrombotic events are observed in the general population at a rate of background, that is in the general population, 2 to 15 in a million people. 20% of these venous thrombotic embolisms are in cancer patients. In individuals who take drugs, that is oral contraceptives, 63 per 100,000 or 630 per 1 million women might develop clots. In pregnancy, 85 per 100,000 or 850 women per 1 million pregnant women can develop this condition. With COVID-19, 25% of COVID-19 ICU patients are likely to develop clots. Last but not least, with SARS-CoV-2 vaccine, with the AstraZeneca vaccine, 222 in 34 million individuals have so far been tested positive for clots. Pfizer, 35 in 54 million. Moderna, 5 in 4 million. J&J, 6 in 7 million have died from SARS-CoV-2 related cerebral sinus thrombosis. So a little bit about the approved drugs, Remdesivir and Dexamethasone. Remdesivir works by interrupting production of the virus. The coronavirus has a genome made up of RNA. Remdesivir interferes with one of the key enzymes the virus needs to replicate the RNA. This prevents the virus from multiplying. Remdesivir, as indicated, should be taken earlier in the disease process. On the other hand, glucocorticoids inhibit the virus through multiple pathways. They inhibit the virus's ability to enter the cell by decreasing expression of S2 and TMPRSS2 proteins. They also inhibit the virus' ability to replicate once it is inside the cell. Glucocorticoids increase neutrophil activity at the injury site, but inhibit further migration of neutrophils and fibroblasts from blood vessels into the site of injury. This means that there is a decrease in cytokine production, and therefore cytokine storm does not occur, neither does scarring of organ tissues. Glucocorticoids should be used with caution because it is associated with suppression of the adrenal axis, which leads to decreased immunity and development of autoimmune disorders. In COVID-19, overuse of dexamethasone has been associated with severe rare fungal infections known as mucormycosis in the eyes, nose, lungs, and sometimes brain, especially in people with diabetes. This condition has led to death of COVID-19 patients who are recovering. Vitamin D has been touted as a COVID-19 protective hormone. It's known to boost the immune system, and therefore it's been postulated to have immune protective effects in COVID-19. However, it has not received any approval from any agency. Multiple studies have been conducted to date. Vitamin D confers both innate and adaptive immunity. Initial evidence dates back to the mid-1800s and the early 1900s, prior to the discovery of antibiotics. Vitamin D was then used to treat tuberculosis. Vitamin D is derived from dietary sources such as milk, fish, egg yolk, and mushrooms. It can also be synthesized from cholesterol by the action of sunlight on our skins. Both dietary and sunlight-derived vitamin D precursors, argocalciferol and cholecalciferol, are converted into 25-hydroxyvitamin D in the liver. 25-hydroxyvitamin D is then converted to 125-OH vitamin D in the kidney or in immune cells, and this is the active form of vitamin D. The optimal recommended circulating levels of 25-hydroxyvitamin D in blood are 30 nanograms per ml. Below that is considered abnormal, and it should be boosted with supplements. Likely infections lead to increased conversion of 25-hydroxy-D into 125-hydroxyvitamin D in macrophages. Remember, macrophages are the resident defense cells that are present at every tissue site in our bodies, that they are the first protective line of defense. 125-hydroxy-D enhances antimicrobial activities of macrophages and monocytes via the vitamin D receptor signaling, which activates proteins that are known as catholycidins and defensins. Catholycidins and defensins are involved in killing microbes by disrupting bacterial, viral, and fungal cell membranes and or disrupting their energy metabolism, or how they derive energy for their activities. Vitamin D also decreases mucus secretion, neutrophil and eosinophil movement from capillaries into lungs, and hence protect against acute respiratory distress syndrome. Separately, vitamin D and its metabolites protect blood vessels from injury by cytokines. Please note that black people or black skin doesn't make vitamin D efficiently. So generally, people who live above the equator or below the equator generally will need vitamin D supplementation. It is recommended that you get tested for levels of vitamin D in your blood so that you can know if you need supplementation. It's also known that people with diabetes, asthma, hypertension are associated with low vitamin D, and therefore they should receive supplementation. Last but not least, the way you ingest vitamin D is key to whether it will be efficacious or not. So currently, bolus doses are not effective in protection. However, daily and weekly doses have been correlated with improved outcomes. There are no regulatory guidelines that determine how much vitamin D is adequate for anybody. However, 1,000 units per day, 2,000 units per day, and up to 5,000 units per day have been shown to be protective and without side effects. Other notable drugs used for COVID-19 include Ivermectin and cannabidiol. Although they are both not approved, they are in clinical trials in the United States as well as globally. In vitro studies suggest that ivermectin acts by inhibiting transport of the virus into the nucleus, preventing replication. Separately, ivermectin may also interfere with SARS-CoV-2 entry into the cell. Ivermectin inhibits other viruses, e.g. dengue, Zika, HIV, and yellow fever. Ivermectin has not received emergency use authorization for COVID-19, but is still being tested. Cannabidiol is derived from hemp or marijuana and is in clinical trial as a therapeutic agent for the treatment of COVID-19. CBD's or cannabidiol's postulated mechanism include increasing endogenous endocannabinoid tone that is known to be protective in the body. Cannabidiol also inhibits cytokine production and migration from the alveolar space into the capillaries. This therefore means that neutrophils will not be activated and they will not try to move from the capillary back into the alveoli space. So hence, cannabidiol will inhibit movement of neutrophils from capillaries into the alveoli. Cannabidiol also decreases cannabidiol decreased neutrophil movement also means decreased cytokine storm, decreased reactive oxygen and reactive nitrogen species, and subsequent tissue scarring. As I end this presentation, I would like to remind us all that it is better to be safe than sorry. Until you are vaccinated, continue to observe COVID precautions. Always check on the conditions on the ground where it is you're going. Do not forget your mask, hand sanitizer, or soap and water if you cannot find a hand sanitizer. As much as possible, avoid indoor environments. If you must go indoors, spend the least amount of time required to accomplish your task. Maintain 6 feet or more apart in the indoor environment. Wear your mask during the entire time. Ensure the mask is covering your nose and mouth at all times. A normal mask is sufficient for majority of the people. If you are in a high-risk group, e.g. diabetes, hypertension, please wear a medical mask. That's the N95. Although not indicated, you can wear protective eyeglasses. After all, for individuals with compromised immune systems, the eyes may be vulnerable to pathogens such as SARS-CoV-2. Thank you for your time, and please leave comments below or questions and we can address them in a later video. Thank you.